in case they want to do it next year. What? Then maybe I'll just go down there. There we go. Good enough for me. All right. Hello, everyone. I'm James. I'm a fifth year in the neuroscience program, about to be in the sixth year since August kicked around. And I am really into open science things, which is a broad term that focuses on getting people to be able to do their research in a way that is transparent and reproducible, meaning that I want you to be able to take your research and be able to share it with somebody else and have them be able to get the exact same results that you did. All right. And in addition, what a brain hack is, I've been to a few, and what we want to encourage for brain hacks are collaboration, idea sharing, and doing something that's cool and that you're interested in. So a little bit before, I'm just going to talk about really brief history of the brain. Super brief. In fact, I'm just going to cover one event. It was time and place long ago, where it was really hot. Not last week, but even further than that, when ancient Egypt. And there was a physician named Imhotep. And Imhotep was one of the first people to actually write down his case recordings of people with brain injuries. So I guess there's a lot of workplace hazards in ancient Egypt, and Imhotep was pretty much the go-to guy for getting fixed up, or at least getting written down so that you could tell what the heck was going on. So in order to write things down, he had to have names for different parts of the body. And before this time, there wasn't a written word for the brain. So he had to come up with his own symbolism for coming up with brain. And this is what he came up with here. And this was the very first actual written recording of the word brain. So now we have thousands of years of forward history coming from this ancient Egypt, and we went from being able to write down the name of the brain to calling it what it is to what. So we've been able to first say the brain is important for things, because before we used to think that maybe it was the heart that was the center of our conscious, then maybe it was the pineal gland in the brain, so just a very particular part of the brain. But we've moved on to a slightly more nuanced understanding. But what have we actually learned? So there's still a lot, but there's still a lot that we can do, and lots of things that we need to understand about the brain in order to really progress the field of neuroscience and adjacent fields, such as artificial intelligence and all these other applications that neuroscience and other of these data problems can relate to. So you might have heard of Neuralinks getting a push from Elon Musk recently, and there'll be other exciting avenues and adventures that people are going through in order to try to really understand the brain at a deep level. But I think that we are at a unique opportunity in time to really hit these questions hard and hit the ground running through through the advent of these online tools and data sharing practices that are now becoming more and more popular. And so being able to combine domain expertise with the knowledge of these tools, I think, will be essential and instrumental for bringing us into the next step of open research practices. So with that said, I'm kind of motivated why we want a brain hack. So now let's talk about what goes into a brain hack. This image is taken from the original publication for on brain hacks, and they essentially have three components where we have a hackathon component, an unconference component, and an educational component. And I'll talk about those three in turn. Where the hackathon is, if anyone's ever heard of the hackathon before, I guess your typical hackathon is a bunch of people that get together in a comfortable space that looks something like this, and you sit on laptops and you code for like 48 hours straight or 72 hours straight with like no rest. They encourage like horrible working habits. That's one thing that's going to be different about this brain hack. Like, as soon as around 5:30 rolls around, like I'm going to be encouraging people to start to clear out and take time for yourself, because I believe work-life balance is very important. And yeah, even though like we're here for like a short period of time, I believe that if we work smarter and not harder is the general takeaway. So for the hackathon portion, we still will be emulating that type of format where we aggregate around projects that that some of you had ideas for, and we'll be giving just like a short little pitch about shortly, so that you'll be able to just tell the group like what you want to work on, and then we can aggregate into those groups based on interest and share to start to like dig into like what needs to be learned about or what needs to be done in order to complete this project, or just like how can we even start to define the parameters of the problem that we are working on. So there can be lots of things that come out of a hackathon. It doesn't need to be code. A lot of it can, a lot of the important work is actually not code, where you're actually trying to define what is the actual question that we are asking, and what and how should we decide on, say, how should the data be organized? What type of users are we trying to accommodate here? Like, where do we find the data to begin with? What already exists out there? Just having like that nice survey at the beginning is going to be really instrumental, so that you don't reinvent the wheel, and B, just write something that you're not very sure is going to do anything at all, and then work in like a not really an organized mess. So we'll have, with that said, I still want you to be able to express yourself creatively. I think a hackathon is about creative problem solving. So just thinking, which an instrumental part of that is trying something and failing. So feel free to try and fail all day, because that is what we're here to do. Then at an unconference session, if we have sufficient interest over topics that we talk about during the day, where people want to, oh, I'm interested in X, Y, or Z. I wonder if like anybody has like some expertise just to like give a little talk about it, or just have some short presentation on it, just so that people can learn. And I think just from conversations around the table, there might be some interest in genomics and stuff like that. So having like a little conversation about just if not enough is communicated through just like interpersonal interaction, we can have a little unconference session that will just give like a really brief overview over like some of the genomic stuff that people want to work on or need to understand in order to be productive. And now, I didn't put a slide in for education, but the education component, will be a couple tutorials. I'll have a tutorial this afternoon and then a tutorial tomorrow morning. This afternoon we'll be covering Docker and Singular, or mostly Docker, but I can touch on Singularity. And that is getting at my goal of being able to essentially package up your research and be able to share it either with yourself 6 months in the future, or somebody else across the world, or somebody across the hall. And make it so that if somebody disagrees with your particular method, that they'll be able to actually test out their chains themselves instead of just saying that I disagree with their method and just saying that's it. So actually being able to have like these enabling like these more productive conversations around around methodology. And for the tool that we're going to be using for communication throughout the Brainhack is Slack. Slack is a really useful communication tool. It kind of fits between email and just like a cross between email and instant messaging, where you don't have to have all the formality of an email and having like these long threads where you want to have this many people for this thread and blah, 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 and it just becomes unmanageable. So Slack is nice. You can make groups and you can make new channels for specific topics that you want to talk about. And it will be a lot easier for us to communicate, and won't have to be in the immediate area in order to effectively communicate. So if you need to have the invitation for the Slack group, you can come up and ask me afterwards, and we'll get you set up to sign up for Slack. And then finally, logistics, just generally for the Brainhack. We have the activity spaces. 5N12 is here. This will be the presentation room for what I'm doing right now. And at 1 p.m. today, we'll also have a company coming over, Flywheel. They do neuroimaging data storage stuff. So they'll be coming and giving a presentation on that at 1 p.m. But other than that today, we can use this space as an open hacking space. As well, the next room is N504, which is just down the hall a little bit on this side. That will be the tutorial room. So starting at 2 p.m., I'll definitely be in that room to help people install Docker on their laptops if they want to participate in the tutorial. And then at 3, the actual tutorial will start. So I've now installed Docker on Linux, Windows, and Mac of different ages. So hopefully I've worked out all the idiosyncrasies of installing Docker on different people's computers. And the ideal is that as soon as you install Docker, then that's the last piece of software that you have to painfully install. And then everything else should be a little bit more of a breeze. And then the rest, or we have N502, which is another room that's just adjacent to N504, and it just has a long table with a TV at the end. So we can use that as the unconference space if the need arises. Such otherwise, we can use it as an open hacking space. And then the rest of the open areas that you'll see around here, we have also some 6-person collaboration rooms along the sides that you guys can use for collaboration. And a lot of the nice chairs that are out here and over there, and some nice couch spaces in case you guys want to sit somewhere a little bit more comfortable for a little while. Those are all available for us to use. And then restrooms and water fountain are outside this space. So outside the door that you came in to get into the informatics center, you take a right past the elevators and take a right again into a small hallway. Then you should see the water fountain and restrooms over there. And I actually haven't been to that space, but there is a lactation room in the lower level room S042. So if you need access to that, I'll be happy to try to wander around with you to figure that out. And then outlets, we do have, looks like people have already found the outlets that are on the tables, and there are outlets along the walls as well. And I asked some of the committee members here to bring power strips. So in case we run out of outlets, which I don't think will be a problem for right now, we will have access to additional power strips to make sure that everyone stays connected. All right. So with that, I want to say thank you to the Brainhack committee. There was a couple of people that had to drop out but did participate last year. Tian had to drop out of the committee this year, and then Hans also had to drop out of the committee this year, but he was an active participant and supporter last year as well. Then special thanks to Marco, who is here in attendance, Gail will be here, and Trevor, who's also in attendance. Without their help, I would not have been able to make this Brainhack the way that it is today. And then I'd also like to thank the University of Iowa Neuroscience Institute. They provided the funding for the food and everything. They didn't make the food choices, so if you have complaints about the food choices, that was me. They escaped money, so don't blame them if I made poor choices. And then UI3, which is they donated the space and a lot of the logistical help, so we're getting things set up. And finally, the brainhack.org, which provides all the information and how to set up a Brainhack yourself, and they provide a lot of the starter materials for that. So I'd like to thank them for just originating this idea and making stuff like this an idea. All right. So with that, we're going to start the icebreaker. So this icebreaker is going to, I'm going to have to look at my name real quick. Yeah, this icebreaker, some of you may have seen this before, but it's a really fun way of challenging yourself to write towards a broad audience. So since the main idea here is collaboration, being able to communicate with other people from different departments and different backgrounds, I think just starting out, we're going to give a little challenge for you to be able to write out what you do for your research in using the 1,000 most common words in the English language. So I'll show an example of this. And if you're already part of the Slack group, I gave a link to the presentation, so you can find this link. Otherwise, you can just type out this link on your browser to go to this XKCD simple writer. I'll give an example of what it looks like and what we'll be doing. So if I was to write down what I researched, and I'm not going to do this on the fly. I already had some time. So I was going to say, I study how brain areas talk to each other while a person is thinking and doing things. So that is about the level of detail and specificity that we can go into with the simple writer. Then, if you found that to be simple, you can talk about say like another hobby or something to add. So I'm going to set a timer for 3 minutes for you guys to work on your statement, your research statement. And then after 3 minutes are up, I'm going to have you find 2 other people that are outside your department or outside your program. I guess neuroscience is a little bit weird since we can kind of be in the same area but talk about totally different things. So I guess if you find another neuroscience person that does something totally different, then that also counts. But find 2 other people that outside your department and tell them your basic research statement using the 1,000 most common words, and we'll assess how understandable it was. It'll be a good way of getting you guys to learn about what each other do. So I'm going to start the clock. The timer here. And it's going. There we go. It's a bomb.
Yeah. So if it's in red, you can't. Exactly.
Yeah, yeah.
If you talk about the other side, then you get to hear about our economy. Inherit is not in there. Yeah, it'll look how they read it. It's not Yeah, I like it.
30 seconds.
Terrible alarm.
Yeah. All right. Who has a complete sentence? All right. So I can give about 50 or 30 more seconds so you can finish up. But now you can read over your sentence, and what we're going to do next is that we're all going to, you can take your laptop with you, but you're going to stand up and find somebody from another department and introduce yourself and give your research statement. Maybe about 30 more seconds. Okay, ready or not, here we go. So can I have everyone now stand up? You can take your laptop with you. And now the minimum is that you can't talk to anybody that was inside your research department, and if you want to do an extra challenge, do something that you haven't introduced yourself to yet. Then we'll reconvene after you guys have finished. So now go around, introduce yourself, and tell about your research topic.
Hi, I'm Maria. Me too. Me too.
Right now, that is psychology. Most of my work I'm going to do my research.
That's why I had people write down their research ones.
Hello.
I didn't really give you enough time, actually. Do you have like a short speech? You can take it out.
All right.
What was yours? And that was a good one. Mine was also. I studied I said also like to teach others how they discover new things in computers because others can do the same thing. Apparently that was an excuse I had to rely on things. Yeah.
Yeah.
Continue for about another minute, and we'll wrap up again.
My younger brother is, he's also he really loves finance. The decisions are good. People don't, if you don't want to take a chance, then a lot of people and usually you don't know what they're doing, why they want
We're gonna wrap up our conversations and bring it down to the table.
And I had like, yeah, so
Oh, you just got a score. Yeah. 121, you said? Yeah.
So it's like
Yeah.
I do.
Yeah, things can move dramatically every year. That's what I tell myself at least. I have a great thing. So we'll see. Yeah. It's like a quick
I was like, if I seen you guys, I know your parents are
Yeah, we do. You can have the HDMI to Okay.
Yeah. Sorry. I can't see.
All right. I think we're gonna circle back up the discussion here for a minute. All right. So, does anybody have research statements that they would like to share? Or does anybody think that anybody else's research statement should be shared?
Maria's. Call me out. I can share. So I'm Maria. I study brain metabolism. So what I wrote was, I studied how the brain makes and uses power. The brain needs a lot of power so you can think and move, but it doesn't have a good way to store power. When the brain gets given, when energy or power that gets given to the brain doesn't match what the brain uses, this can make the brain sick. I study changes in different kinds of brain food used for brain power in different kinds of sick brains.
Wow, that was like a
That's
paragraph.
See why I suggested it.
All right. Any others? It doesn't need to be. It can just be a sentence long too. Don't let that be. That was good. That was really good, Maria. Yeah. Does anybody else have a research statement that either they want to share or somebody else's that they really like to hear?
I thought Trevor's was really good.
I am studying how brain areas talk to each other after working out, especially as it has to do with sleep and normal body moving.
All right. Awesome. Yeah, that's a great one. Any others?
Liked Marco's and just project. Sorry, terrible names.
All right. Do you guys mind sharing?
Marco.
I should delete mine.
Where's the window?
I just wrote a beautiful sentence.
Yeah. Do you want to share your partial sentence?
I study brainwaves.
All right.
So we studied neural oscillations and how the thalamus uses them to coordinate with other parts of the cortex or other parts of the brain to achieve cognitive control.
All right.
So I didn't know how to write that.
Yeah, no, this is a very difficult exercise. Yeah, it's trying to like condense down all the years of your learning into a very small and limited vocabulary. So it is a very challenging experience. But I applaud all of you for taking on this challenge.
It doesn't even like teenagers.
Yeah.
Yeah. Even you can't even support teenagers. Gosh, yeah, it's very difficult. Okay, so with that, the goal of the exercise was to try to condense your complex research topics into a simple language so that you could communicate it to somebody that was outside your department, so that you'd be able to transmit some information from what you do into something that they could understand. So I hope that was an outcome of the exercise, and also to get you guys to introduce yourselves and get to know each other a little bit more. So I hope that that was successful as well. So with that out of the way, we're going to move on to the next item, which is going to be the project pitches. I have the proposed order in Slack under the announcements channel. So I guess I'll ask now, has everyone signed part of the Slack group? Razor, you had trouble?
Not, yes.
Okay.
So, I mean, it asked me to check my email. I sent it to my email and nothing came to my email.
Check my name.
Did you check your spam folders?
You got it.
Okay. Then,
Oh, here it is. I literally
okay.
Awesome. Yeah.
I've already taught you guys.
Okay. So anybody else? Did you have trouble?
Yeah, I got it.
You got it?
Yeah.
Okay. So you're able to pull it up. Anybody else have trouble signing into Slack? All right. I guess maybe I should answer it the other way. Who has successfully signed into Slack? Raise your hand. Got it. Okay. Then did you raise your hand? All right.
Yes.
Okay. I think we're all good. If my eyesight does not deceive me, or no? Is somebody missing? Ethan?
Yeah.
Okay.
Yeah, I'll give you an update.
Okay. Let's status update.
Can you use mine? Yeah, there you go.
Okay.
There you go.
So the first project, I'll give a little presentation about something that I'm working on. I don't know. What if it's going to be the top hit on Google now? or Bing. I can't believe I'm using Bing. All right. Okay, so for the first project, this is something that I've been working on for a while now, and I can take in people from a variety of different coding backgrounds in order to try to fit a challenge that meets your skill level, or your current skill level. And the name of the project is NiBeta Series. What it is, it's a piece of software written in Python that helps calculate what I said during my research statement, kind of how brain regions talk to each other, or specifically, how do the activations in those regions correlate with each other across different trial types. So as somebody views a screen and sees a picture of, say, Daniel Radcliffe versus Elijah Wood, maybe there's two brain regions are very in sync for Daniel Radcliffe, but maybe they're really out of sync for Elijah Wood. So I'm looking for how those networks emerge, and where can we find those networks in the brain using this software package. So with this, I still need, there's still a lot of things that could be improved in terms of usability. There are documentation fixes to make it clearer. And there are other things I would like to implement to make the package more fully featured and to accommodate a very popular data standard that has come out of the neuroimaging field called the Brain Imaging Data Standard, I guess or BIDS for short. So if you want to hear more about BIDS and NiBeta series and those types of things, and you want to say, I should have a couple issues in which if you've never worked with GitHub properly before and want to submit like your first pull request, I can help you get started with that. So that's all I wanted to say about NiBeta series. And next will be Michelle. And then you'll connect your laptop, and I'll try to press the right button.
Did you guys introduce each other to everybody?
What's up?
Was the icebreaker the first introductions?
I'll say, yeah, we didn't go around the room and like introduce ourselves, but yeah, you should introduce
Of
yourself.
course I'll introduce. Let's see if I was behind. Apologize, I was late. So always has trouble.
Like, yeah, the screen's not.
And then you swap and swap and swap.
There we go.
All right, so I'm Michelle Voss. I'm in Department of Psych and Brain Sciences. So I run the Health, Brain, and Cognition Lab. And Trevor is a student in our lab. So his introduction to research is a lot about what we do. We study how physical activity can impact the brain as we get older. You might have antenatal experiences. Cognition tends to get a little slower, particularly memory declines. And we're interested in ways to prevent that as you get older. And we want to study how it works. So we use neuroimaging. James is in our lab. And I've never done a project pitch at a Brainhack, so I wasn't sure what to do. But I wanted to introduce you to this package that I've gotten really interested in and really impressed by it. And it reflects a toolkit for doing functional connectivity analysis that I think reflects the way forward in terms of it's really well developed, it's online, it's in an open source framework, and it's extensible, meaning it's got the base there and you can extend it, and we could extend it. So what is functional connectivity? It's a broad audience. I figured I would give an example of our research and why I would be interested in looking, or why one would be interested in looking at functional connectivity. So this is an example of an inflated human brain. All right. And these little dots would be regions that, based on some literature over the past about 10 years, I would say, people have developed, let's say, an idea here then that these regions are really important for being able to remember our experiences in everyday life, and the different colors would denote different sort of subsystems in this memory system, this broader memory system. So we have the hippocampus here, and then my daughter wanted to paint nails last night, so she picked blue for me. They actually match. So we have the visual cortex, the hippocampus, and the prefrontal cortex, and these are regions, the parietal and prefrontal in particular, are areas that actually sort of like disintegrate. I don't know, that's a really strong word, but fragment away from this area that's really important for memory. So we like MRI, or I like MRI a lot, because it's a method, it's the only method that you can look throughout the whole brain at once in the same individual, measure many different aspects of brain energy, sort of metabolism, flow, many different things, structure, all in the same person at the same time, right, throughout the entire brain, deep structures and cortical structures. So there's a lot of power there. And this would be an example then of regions that we might be interested in how they work together to accomplish memory, because we know that when things break down with aging, it happens to be a lot of breakdown in networks. So these are just the different labels that people give to these different subsystems. And to give you a sense of what that data, how that data gets created, or how those claims get created, you can imagine each one of those circles is a region, right, that I said, and you give labels to them. And this would be a cross-correlation matrix of the bold signal in the scanner and how basically the signal correlates over time. So a brighter color would say that the regions have a really similar time course, and a strong time course in the bold signal is about 0.65. So those would be two regions that are pretty well communicate. Well, it's claimed that they're communicating because they're going up and down at similar times over time. Okay? And you can see the bold signal is really slow. And so two regions that would be claimed to be in the same network or same system would probably have a correlation of 0.65, whereas two regions that are sort of more weakly connected but also claimed to be in the same system would be about 0.3. So weaker, stronger, but still part of the same system, whereas a 0 would be claimed to be not interacting at all. Okay. So what we would do with this data is we would take it from an individual. We would take signal over time from an individual. This is one person, one scan, but we want to go beyond that. And we want to say on average, what was their connectivity, their functional connectivity, and then do things like average across regions in different systems. So let's say this is the red system, we want to get an average value, and this is the blue system, we want to get an average value. And a lot of things that happen with aging that go south are to do with specificity. So we might want to look at cross-correlation between systems, and it should be low. Okay. So that's what we'd want to do with it. And what we've done so far is we use an in-house script library that we have developed over time that I first cobbled together in grad school with no background in programming. But this XCP software package is developed by a really strong group of people that care a lot about one of the biggest issues in MRI, which is motion. So it's a third variable. If the head is moving, you're going to get correlations over time. So it's a big issue, and people that have autism or any other psychiatric disorder that some of the people in the room might be familiar with, they also move differently. So motion's like a really big issue, and this group really cares about it. Older people also happen to move more. And like I said, they have they reflect, I think, what the future will look like in terms of documentation. But right now what they're doing is they're giving you the basis for giving you basically the functional connectivity in its rawest form, which is per person, per link. And what I would propose is we work on just taking that output and creating files that give you per person an average on however you want to average different nodes, and really flexible. And I would propose we work in R, because that's my personal favorite. But I'm open to whatever, and as a disclaimer, I'm not as savvy on GitHub as maybe others are. So if anyone were to be on the team, we would need a better expert in GitHub. So that would be, I can put this on Slack. But
All right. Thank you,
I
Michelle.
don't know if there's questions or
Yeah, we can open the floor to any questions.
James or.
Have you used it at all?
I have used it. I have a starter notebook for taking the output from 3 people to get us ready for the CREST data set. I have a starter notebook. And so we're working on analysis, for example, with CHUSA, where we want to look at the functional connectivity of a system with a different parcellation, which is just how you divide up the brain into different regions. And so I've started an R notebook on averaging across the different networks. Yeah. And it's really fast. It's very fast. And it's really, one thing that I love about it is it makes it very easy to run different iterations of different time. I'm going over time. Going over different denoising strategies.
Yeah, it's a super nice tool.
It's really fast.
Yeah, so if you have more questions about that or you're interested, I think it would be, I think we wanted to learn a little bit more about, say, R and or Python. This would be a great opportunity to learn like how to read in files and how to do some manipulations to like a tabular file to switch it out from, say right now, a very long format to perhaps a wider format, and doing those types of data manipulations. All right, so next I have Taryn. Like you don't have to.
It's actually
like
Leo.
a big
This is
presentation.
most
Is it Leo?
I just wrote the summary for it.
Oh, you just wrote the summary for it?
Yeah.
Okay. Yeah, like I said, it doesn't need to be anything big.
Yeah, okay, that's good because I don't have a presentation.
Yeah.
So I'm Leo, for those of you I haven't met yet. And so today we're going to be working with a lot of brain MRIs. The general question is going to be, how can we improve the generalizability of machine learning models trained on one brain MRI data set when they're applied to a different brain MRI data set. So the thing we're going to be predicting is actually just like sex. So ahead of this, I downloaded around 4,500 brain MRIs from 100 different sites, like across the country, I guess, probably world, I don't know. And so we're going to be doing deep learning. So we're going to be training like fancy models. They're called like convolutional neural networks, which we could talk about that at some point. But we'll be training these models and we'll be predicting sex on, say, like, we'll use maybe 3,000 brain MRIs from one part of the world, and then we'll try to transfer this model to the remaining MRIs and see how well like the model performs. Did we get a decrease in performance? And to try to see if we can increase the generalizability of this model, we're going to take 3 different approaches. So one is that something probably many of you would be familiar with is like pre-processing your brain MRIs. So in the first case, we might just use like sort of just the raw data that these different sites have given us. They're not like registered. They're going to have different brightnesses. But I've gone through and processed them. So I did registration and brightness stuff. Not really my specialty, but so some of it's, that should hopefully improve generalizability. Another thing we're going to do is technically called like data augmentation is what it's called. But essentially, when you're training a model, you apply all these like weird transforms and you make it bright and dark, and so you add noise to the data, noise that might typically be found in like one imaging site versus another. So by actually introducing yourself, it's going to tell the model not to learn that, and that should hopefully increase the generalizability of the model. And then the last one's like really hard to explain, but it has to do with the actual machine learning model itself. You give it the thing that you don't want it to learn, which is the site that the image is from, and you say like, don't learn this. And there's like a complicated mathy way of doing that. So we have these 3 different approaches to make sure that the models will generalize better, and we're essentially going to be benchmarking them to see how well the models perform. So it'll be a lot of like using Argonne and running deep learning models and working with brain MRIs. So,
I have something to show you for working with Jupyter notebooks on Argonne. So if you want to see that, we can talk.
Yeah.
Having it so you don't have to do like the 3-port forwarding or whatever.
is it the like the Kingly?
Yeah, the Kingly. Do you already? I guess
we have that, but we have to. I mean, they're like really big, so we have to use the full like GPU.
Okay. Okay. All right.
Yeah.
I have a question.
Yes.
So what's the age range of the?
Yeah. I haven't looked at that. It's big. We were gonna predict age, but then I wanted more images. So some people like release brain data sets and they give you like no metadata. And so if I used age as the thing we were predicting, then we would only have like 3,000 images. So I haven't looked at the age because we don't have the age. Yeah, it's gonna be highly variable.
So, but those include like adolescents and infants?
Yeah, I mean, so like the data that I used is like Open Neuro is one of them. I use 2 autism data sets, so that's gonna have like a ton of people who are like 9 and 10-year-old. Yeah, it could be like the full range of ages.
Because I read a book called Pink Brain, Blue Brain, and the argument is that the infant's brain look no significant difference between boys and girls.
Well, that will decrease our performance.
So I think like age may be a big issue.
Yeah.
If you also include infants.
Luckily, we can't look at it, so we don't have to.
It's already constrained.
Yeah.
Right.
All right. Then, any other questions? All right, you're off the hook. And next, I had Ethan.
I don't have any slides.
Yeah.
Or a written summary.
Yeah.
Or really anything at all. But.
And it's just supposed to be informal, so that's good.
Yeah. So I am doing some genomics work today. I will be working with Leah, our visiting scholar from Germany. And what we are trying to do is, well, let me back up. So we know that whenever your brain is active and different regions are communicating with each other, processes like synaptic plasticity allow your brain to change over time. And this is highly dependent on gene expression. And the way that that gene expression is coordinated in response to brain activity requires regulatory regions of your genome. So not genes themselves, but other regions out in the middle of nowhere in the junk DNA. And so we have some data sets that have looked at the actual proteins that bind to these non-encoding regions of the genome. And so we're trying to model DNA sequences to see how, as you change a DNA sequence, how does that change its capacity to function as a regulatory element? So we're going to be training deep learning models like Leo was mentioning, although most of what we'll be doing today is guaranteed to just be data gathering and training data generation. So, any questions? All right, well, come find us if you're interested.
All right, awesome. Thank you. All right, next I had Maria.
Yeah, I have a presentation.
You do have a presentation. Okay. What type of connection?
It's HDMI. It's just weird on this end.
Okay. That looks like that's good.
Thank you. That was awesome.
Okay, well, most of you have already kind of heard what I talk about from my great elevator talk. But kind of more broadly, my lab is studying metabolism. And so we've done this like looking at animals, how their whole body uses energy. And then more recently, we've been able to get omics data. So kind of related to the genetics work that's been mentioned already. We're able to gather a pretty wide platform of different metabolites, and just things that are broken down to provide energy. Specifically, I'm looking at the brain. To kind of compel you that this is interesting, energy mishandling is really important. You can see it in a lot of, especially diseases in developed countries, so things like obesity, diabetes, heart disease, and then of interest to us, neurodegeneration. And so we're trying to figure out what energy changes might actually be driving these diseases, and if we can identify them, can we treat them and target them in other ways? I'm really glad that Michelle showed that image of kind of the node that you can see in the brain with connectivity. I don't know if you can really decipher this very well, but this is a metabolic pathway. If you know of the Krebs cycle or the citric acid cycle, it's this little bit right here. The rest is just a bunch of different processes that actually go on in metabolism. And this is what we're trying to model, what we're trying to look at. And you can see these different colors and these different little nodes are representing the different metabolites and the different networks that are occurring in these different pathways. And so these are different things that we're trying to model and look at. This is just a representation, you don't have to memorize it. In general, yeah, right. So what we are working with, in my hands, I work with either cells or animal tissues, and I extract metabolites from these different tissues, and then we use different processes such as gas chromatography and liquid chromatography coupled with mass spectrometry to break apart these different metabolites and identify them and quantify them. And so that's what we're getting for our data, and then the output that we get are either around 100 different metabolites or 200 different metabolites depending on the different machines that we're using. This is kind of like, it's much smaller, of course, than RNA-seq and things like that, so it's kind of like bite-sized metabolomics and something that we're working with right now. And specifically what I'd like to do, we do have a variety of different online tools that we've found to kind of work with this, and we're just kind of piecing them together right now and using a lot of Excel spreadsheets to process our data. And so what we get is we get our data output back with all of the different metabolites for all of our different samples and our different groups. And we have to correct for machine drift during the experiments. We normalize to our starting material content, we remove outliers, and we normalize. And then after that, we perform our analyses. And so my dream would be to just make some sort of code that can do all of this in one hub instead of going from one website to another and then plugging everything into my Excel worksheet. And so if we could maybe talk over some things like that, if you would have any ideas. I know nothing about coding, so that's my disclaimer, but it could be a kind of cool little project, I think. I think that's all I have.
All right.
Does anyone have any questions?
Can you go back to the last slide?
Yeah, of course.
You mentioned machine drift. Is that data from the mass spec and the liquid, the gas chromatography? And are those values that are that have been recorded like each day? Because I mean, I'm aware that with MRI, you the actual field strength does change, and you have to account for that when you're shimming the magnet and all that. But are those values that the people who run it give to you?
Yes. So the machine will take around maybe like 6 hours to run all of our data or our samples that we'll put into it. And during that time, we'll have these pooled samples. So we take a little bit from each of our samples and have internal standards in them. And we run them at various intervals throughout all of the other samples being run. So we'll do like our experimental groups flanked by these internal standards. And then over time, we'll correct that and see if like our internal standard is fluctuating. That means the machine was doing something slightly different then. So that's what we're correcting to. And I don't know how we use this online program for that. So I'm not really sure what it's exactly doing to correct for it.
Okay.
But that's the basics of what it's doing. Yeah.
So your project, what do you start with in your project, and what's like the end goal?
Yeah. What I start with for data is an Excel spreadsheet that has my samples and all the metabolites and the internal standards, and then I do those corrections. And my end goal are like, I do a lot of ANOVAs, depending on how many groups, I'll do volcano plots and things like that, and heat maps and PCAs, that kind of thing. And then we're also trying to do a little bit more elaborate is looking at pathway analysis, so kind of looking at those different hubs, seeing if they're different, seeing if there are like different breaking points of like two adjacent metabolites, there's some sort of disconnect between them when they should be kind of related, kind of identifying those.
So not on that, you say integrate analysis, that's what you said.
Yeah.
A lot of analytical things.
If you have other suggestions, though, I'm open to hearing them. Okay, thank you.
Yeah. All right. Thank you. Was there anybody else that everyone that said on the registration form that they wanted to give a project pitch? Was there anybody else that wanted to give a project pitch? Otherwise, I just have one more little thing. Or one more little pitch in case people are interested. Anybody else? Going once? Going twice? All right, sold to me. So I just have one other small thing in case people are really into coming up with data standards. For whatever reason. I'll pull it up in here. Wiki projects. So what I mentioned before in my first one about the brain imaging data standard, this is a living document that is constantly being extended and modified to fit the uses of researchers. So there's a main specification that kind of gives the how you should name your files, how you should organize your directories, and all that. But they also have a ton of BIDS extension proposals. And if you've ever been a part of the Python community, they have something similar called like the Python enhancement proposals. So a BIDS extension proposal is a piece of data that is a little bit, so if the overarching BID standard kind of gives like a general rule set, then these extension proposals help define specific implementations and give out the little nuances that one needs to consider in order to work with their particular type of data. So given the audience here today, I thought an appropriate one would be BIDS extension proposal 18, which isn't very explanatory until we actually go and look at the title of it. So this is about genetic information. Specifically, how do we tie together say some type of imaging data or some type of brain data with genetic data. So this is helping just define the standard on how we should relate these two pieces of information together in a standard way. And why this is important is as soon as we come up with a way of how we want to relate these pieces of information, that allows people to start to build tools around it and make it easier to develop and maintain those tools. So when we have a solid idea of how we want our data to be organized, it makes people who make tools lives so much easier to be able to make those assumptions about how the data are organized, and then be able just to read it in without having to either just assume some sort of arbitrary organization that then the user has to then modify all their input data to make it fit for that specific tool, and then they have to modify it again to pipe it into another tool and stuff like that. So the idea behind these is to come up with this standard outcome and a standard organization to be able to have greater interoperability between these tools. So if you're interested to read more about this, yeah, feel free to come talk to me. And we can see, I'll post this on the, I'll post this link on the Slack so that you can get an idea of what all the projects are.
I see the pigs up there.
Oh, sorry, I should have changed that one.
I can explain what it is, because if anyone would be interested in it.
Yeah.
We have this project that's starting up that we're trying to test the feasibility of looking at how exercise affects energy metabolism in the body and the brain, and animal model that is a little bit more similar to a human than a mice or a rat, and this would be a pig. And one benefit of this is that in humans, we can look throughout the brain all at once and get blood samples, but we cannot get biological tissue from an in vivo in a human. So we have trouble connecting what's going on in the blood to the brain, but in rodents, we have great access to the brain, but their muscle metabolism isn't as similar to humans as other animal models that could exist. So we're starting this pilot project, and we have one MRI from a pig, and the pitch was just what does one need to do to use some tools like MRI-QC and fMRI prep on a new species that hasn't been used in, and what would be the steps to go forward and look and see if they have similar resting state networks as humans. So, yep.
All right. thank you. Any questions about that?
They're an
Very-
exercise.
It's
We
very
ultimately want to
interesting.
look at metabolomics in those pigs and connect across all levels of the system.
Right. All right, so are there any questions about that project or any of the other projects that we're going on right now? Okay, so if there are no questions, then the next step is to now aggregate yourselves into the group that you would be interested in, and yeah, and if you are feel like that you don't have a group or that you don't have a place that you want to participate, you can come talk to me, and I'll try to find you a group or give you something that can that you can help to work on. I'll probably just recruit you to help me with some of my projects. So that I don't monopolize, you should find somebody else to work with. Okay, so with that said, I'll open up the spaces here and we can get started on